Hi everyone. I just want to start off by thanking the organizers for inviting me to give this talk on some of the work that we've been doing at UC Berkeley, where I'm currently a postdoctoral fellow in the Tooten Lab. And also thanks to all of you for joining in today. I'm very excited to be surrounded by so many color vision enthusiasts. Today I'm going to be talking about the dependency of color appearance for small spots on the spectral topography of the cone mosaic. And so when I say the spectral topography, what I'm referring to is this organization here of the three types of cone in the human retina. So let's just back up a little bit and start with a fact that we're all, of course, familiar with. Normal human color vision is trichromatic with three types of cone photoreceptors that are sensitive to short, middle, and long wavelengths of visible light. So these are typically referred to as the S, M, and L cones, showing the spectral sensitivities of the cones plotted here from the Stockman and Sharp cone fundamentals. And with various cone classification techniques that use adaptive optics, or AO, which I'll be explaining in more detail later, we can actually classify the different cone types and visualize the spectral topography of the cones in different individuals with images shown here. So these are real images of different people's cone mosaics that were gathered from a few different publications using different AO classification techniques. And the spectral identities of the L, M, and S cones here are colored red, green, and blue, respectively. And in showing these images to you, there are really two key features of the trichromatic cone mosaic that I want to point out. And the first is that the proportion of the long to middle wavelength sensitive cones, or the so-called LM cone ratio, varies significantly across individuals. So in these examples, the percentage of L cones ranges from 27 to 94% at the extremes. And the second point that I want to make here is just that the spatial arrangement of the L and M cones is nearly random in the mosaic. So this results in some spatial clumping of cones of the same spectral type. And so a major consequence of this is that there are then these local variations in the proportion of L versus M cones that occur at different locations in the retina. And then the demographics of these local variations are, of course, based on the global LM cone ratio, with more clumping occurring in L or M cone biased mosaics. But we know that despite this variability in the spatial arrangement of the cones, visual system produces largely consistent color appearances. So this leads us to the question that motivated the work that I'll be showing you today, and that is, how does the visual system produce consistent color appearance despite variability in the spectral topography of the cone mosaic? So this isn't a new question. In fact, as we'll see in the next few slides, this is a question that has been of interest for quite some time. But there are two key pieces of information that are afforded by recent technologies that can help us approach this question in a novel way. So first, we need knowledge of how the L, M, and S cones are arranged in the photoreceptor lattice, and which, as we've already seen, it varies substantially between individuals. So we really need to be able to identify what these cones are in our specific subjects. And second, we need control over how these cones that we've spectrally identified sample the retinal image, or more specifically, the stimulus that we're presenting to them. So to tackle the first of these, we need spectral classifications of the cone photoreceptors and the knowledge of where those receptors are located. So this slide here was borrowed from Professor Will Tooten at Berkeley, my postdoctoral advisor. And it's showing how adaptive optics optoretinography is used to classify cone photoreceptors. So AO compensates for wavefront distortions that are caused by irregularities in the optics of the eye, and this allows for near diffraction limited resolution in imaging of the retina. So in the case of AO optoretinography, adaptive optics is combined with OCT in order to measure changes that are occurring in the optical path length of the cone outer segments in response to light. So in this example, it's looking at the response to long wavelength light at about 660 nanometers. So the response curves of the cones, as you can see, segregate into different clusters. And here, the cones that are having the largest difference in the OPL are the long wavelength sensitive or L cone photoreceptors. So this is how we can disentangle which cones have a particular spectral sensitivity. So this technique was first applied in Don Miller's lab at Indiana University. And more recently, it's been used by Sabesan and his group at the University of Washington, where we've been really fortunate to collaborate with Ram and his students to have our subjects' cones classified for the study that I'm going to be talking about today. And I won't go too much into depth in the interest of time about the different cone classification methods that use AO, but one thing to note is that this is not just the most recent method that's being used, but it's also the most precise. So with the knowledge of the cone spectral types and their locations in the mosaic, what remains is the need for control over how those cones that we have classifications for sample a stimulus that we present. So this relies on retinal stimulation with an adaptive optic scanning laser ophthalmoscope, or an AOSLO. And this device can really be broken down into two components. The first of which is compensation for optical perfections of the human eye, which is achieved by this adaptive optics portion of the device. So a wavefront sensor is used to measure irregularities in the wavefront of light as it's reflected back from the subject's retina, and then a deformable mirror is used to correct for the wavefront aberrations that are measured, so that the light coming back from the retina is then corrected optically, allowing for high resolution imaging. So shown here are images from Alf Dubra and his colleagues with the dense cone packing shown in the foveal center, and then the larger cones surrounded by rod photoreceptors in the periphery. So the second piece of the system, the second piece of the system is stabilized stimulus delivery via real-time eye tracking. So this is achieved by the SLO component of the device. And because the light is scanned in a raster pattern and images are acquired in real time, we can actually track the fixational eye movements that are occurring during a single frame scan. And then we're able to update our stimulus timing depending on the retina's current position. So we turn on stimulation light with a fast optical switch when the scan passes over a region of interest. So we can actually target a single cone, or in this case, a small group of cones as shown here, to be stimulated with light that has been corrected by the same adaptive optics system that we're using to image the retina. So with both of these requirements covered, knowledge of the cone types and locations, and the ability to stimulate specific cones, we can next think about how to address the scientific question posed here, which, just to remind you quickly, is how does the visual system produce consistent color appearances despite variability in the spectral topography of the cone mosaic? So here we're looking at the proportion of L versus M cones across a larger sample of subjects. So this was measured with ERG, a technique that's used to measure the total LM cone ratio across the entire retina. But from this, we can't necessarily know where those cones reside. But it is a good technique for comparing LM cone ratio across a large group of subjects. And so as we can see from this figure, the proportion of L cones does vary widely across individuals. But we know that when we ask observers with widely varying LM cone ratios to then identify a color that looks uniquely yellow, that is, it has no hint of either red or green, there's actually no correlation in unique yellow settings with LM cone ratio, at least with stimuli that are about a ½ degree and larger in size. So in fact, unique yellow is remarkably stable across individuals at these larger stimulus sizes. And adaptation studies have shown us that the wavelength of unique yellow may reflect the spectral characteristics of the environment, and therefore it could be completely independent of these anatomical differences that I've been discussing so far. On the other hand, when we think about color vision at the cellular scale, cone signals are ambiguous. So if we target a single L cone with 543 nanometer light, so this is a wavelength where L and M cones are approximately equally sensitive, but it typically appears green when viewed under normal viewing conditions with a larger field stimulus. So in this case, when we're targeting a single cone with this light, the brain can interpret this signal as a chromatically uniform red spot of light based on this difference we see between the stimulated L cone and nearby M cones that might have lower excitations. But an equally reasonable interpretation of this cone activation pattern is a black and white edge. And this is because cone signals, of course, are univariant, and different stimuli can achieve essentially the same cone activations. We know from single cone color naming studies that in this scenario where a single receptor is stimulated, color appearance depends highly on the spectral identities of the stimulated cones. So here I'm showing you a figure from Sabesan et al., where subjects were asked to respond with the color names shown here following a single cone stimulation. So the donut histograms that are surrounding each cone in the mosaic show which cones were targeted during the experiments and also the proportion of responses across all of the trials. And what they found was that L cones, shown in red here, typically elicited red color responses and M cones green color responses, and then a fraction of responses from either cone type were reported as achromatic or white. So the specific question we're trying to address with our work is what happens between these two extremes. At one end, where single cones are stimulated, color responses depend heavily on the spectral identities of the cones targeted, whereas larger stimuli produce consistent color responses that are independent of the relative L and M cone proportions. And one way that we can more specifically frame this question is at what spatial scale do the mechanisms operate for compensation in the difference in relative proportion of L and M cones. So let's return to unique yellow and develop our thinking a little bit more. So if we first consider a typical mosaic with a 2:1 LM cone ratio, and this is just a hypothetical mosaic, in this case, the relative summed L cone signal is roughly twice that of the M cone signal. And if we assume that we want the comparison of the L and M cone signals to have a zero crossing at unique yellow, or really just any point of red-green equilibrium, we would have to scale down the sensitivity of the L cones by some factor here that we'll call K. So L minus M signals that fall above zero would then result in reddish colors, and below zero would result in greenish colors. And importantly here, individuals with different relative numbers of L and M cones would require different scalings or different values of K. And if we think about this within the same individual, if the same scaling factors then applied to every M cone in the mosaic, then a problem emerges when stimulus size is reduced. So if we target this patch of cones here, where there are fewer L cones than the global average, the L minus M signals would be negative when viewing 578 nanometer light, and therefore they look slightly greenish. And a consequence of this would be that if we measured unique yellow just for these cones, it might require a shift to longer wavelengths of light. So the opposite scenario would arise here, where there are actually more L cones than the global average, and unique yellow may shift to shorter wavelengths. So the plot I'm showing here is the prediction from a simple model where every cone in the mosaic is scaled by the same factor to set unique yellow at 578 nanometers for a large field stimulus. The y-axis tells us the predicted wavelength for unique yellow as a function of the local proportion of L cones that are sampling the stimulus. And the local cone neighborhoods that have similarly proportioned cone identities to the global average plot at 578 nanometers. So by contrast, imagine that the mechanisms that compensate for differences in cone ratio operate at a more local scale. So in this case, the prediction is that unique yellow would be invariant with respect to local variations in the spectral topography. And one way that the visual system might compensate for spatial variations in the LM mosaic is by learning that the activity at a particular location in the mosaic in response to some wavelength, say 580 nanometers, is associated with the response of that part of the mosaic to yellow light. And this could perhaps be a similar mechanism to the one that makes the wavelength of unique yellow invariant across individuals with different LM cone ratios. So if we consider a hypothetical mosaic here that contains about 70% L cones, the relative L cone activity at 580 nanometers is several times that of the M cones. However, for a smaller region of the mosaic, where the local L cone percentage is only 30, the summed activity of M cones is higher than that of L cones for the same wavelength. So the global compensation hypothesis predicts that unique yellow will occur when the total L versus M activity reflects that of the larger mosaic in response to 580 nanometer light, perhaps because this information about local spectral variations is lost through a mechanism like spatial summation. Whereas the local compensation hypothesis predicts that yellow arises when the L versus M activity reflects that of the local signal at 580 nanometers. So to distinguish between these two hypotheses, we wanted to measure the topography of unique yellow with small stimuli. So we measured unique yellow for retinally stabilized stimuli that were 2.18 arc minutes in diameter and were presented at 1 degree retinal eccentricity. So stimuli were square and arranged in this grid pattern with 3 rows of 5 stimulus locations. And our experiments included 3 colored normal subjects, 2 of which have classified cone mosaics from our friends at the University of Washington, and 1 subject whose cone types are still unknown, though we're hoping to have this data in the next couple of weeks. So to measure unique yellow, we adjusted the intensities of the 2 primary lights in our AOSLO systems. These were 543 and 680 nanometers. And unique yellow was defined as the dominant wavelength that's metameric to the stimulus mixture. So shown here is an example using the McLeod-Boynton chromaticity diagram with the spectral locus shown as the solid black line. So in this example, a stimulus that was comprised of 40% red light and 60% green light appeared uniquely yellow, and this corresponded to a wavelength of about 586 nanometers. So during the experiment, we used a staircase method with a 2 alternative force choice procedure where subjects indicated whether the stimulus appeared too red or too green on each trial. And the stimulus mixture eventually converged on an estimate of unique yellow. So then we fit psychometric functions to the data that we acquired for each stimulus location in order to have 15 estimates of unique yellow for each subject. So here I'm showing the results with the color overlays showing unique yellow estimates for each stimulus location according to the color bars down here on the bottom. And the cone types for the 2 classified subjects are shown by these small point markers centered on each cone photoreceptor, with red indicating L cones, green for M cones, and blue for S cones. And so these frequency histograms along the bottom row show the same range of unique yellow wavelengths along the x-axis that's shown here in this color bar. And one major takeaway from these data is that there does appear to be a lot of variability in the wavelength that appears uniquely yellow at these spatially restricted retinal locations. And areas of the mosaic with more M cones resulted in typically redder unique yellow responses and more L cones resulting in greener unique yellow estimates. So subject 2021-0R, shown here on the right, had the most long wavelength estimates. So we're really eager to see this subject's cone classifications with the prediction that this subject should have a larger preponderance of M cones in the area of the mosaic where we tested. But we can see just from the raw data here that substantial variability exists for these small stabilized stimuli. And then one last thing I want to point out on this slide is that there were cases where the unique yellow measurements were out of the gamut of the primaries that we had available to us in our AOSLO system. So in that case, we weren't able to estimate unique yellow. So for these measurements, for example, the subject may have said that the stimulus appeared red on every trial. And so the staircase sort of pushed it as far in the green direction as it could, never really reaching a threshold. The last thing I want to point out here is that we can compare this to unique yellow estimates for more conventionally sized stimuli. So since we also made unique yellow measurements with the same primary wavelengths in the AOSLO using the full 1 degree raster scan, so that's shown by this dotted line here, which are plotted over the frequency histograms. And we found that despite the large variability for small spatially restricted stimuli, unique yellow estimates for these larger unstabilized stimuli were in line with prior observations at around 580 nanometers. So the variability that we're seeing already in unique yellow estimates for small stabilized stimuli seems to provide evidence against the local compensation hypothesis, because remember, in this case, unique yellow is expected to be invariant with the local spectral topography. But what we really want to do is have an estimate of the local spectral neighborhood for each stimulus location. So we need a way to estimate the local proportion of L cones, taking into account not just the stimulus size and its position, but also the various sources of blur in the stimulus during the experiment that would engage cones slightly outside of the stimulus profiles that are shown in these images here. So to do this, we modeled the stimulus at 2.2 by 2.2 arc-minute square as a convolution of the stimulus with the AO corrected point spread function, in addition to residual blur from transverse chromatic aberration, or TCA. So we did correct for TCA with established image-based procedures, and then we monitored the pupil position to keep the pupil in the same position with respect to the AOSLO beam, but we do assume that there's still some residual blur from slight changes in pupil position that are pretty much unavoidable at this point. And then the third thing we have to consider is blur from stimulus placement accuracy. So the delivery technique that we use does result in slight errors in stimulus stabilization, but these can be measured post hoc through analysis of the videos that are taken during the experiment. So we apply this modeled stimulus to the cone sampling array, where each cone, L or M cones, are modeled as a Gaussian. And we perform a simple multiplication between the stimulus and the cone arrays to estimate the relative L and M cone proportions responding to each stimulus. So that's what's plotted along the x-axis here, where the local proportion L for each stimulus location is plotted against the experimentally measured unique yellow estimates. And the error bars here are 95% confidence intervals on the psychometric fit parameters. And so from this figure, we can see immediately that the data do deviate significantly from the local compensation hypothesis. And instead, we see that the variation in unique yellow does appear to be dependent on the local spectral neighborhood. So we then compared the data to the global compensation prediction that I showed you before, where unique yellow is achieved by replicating the global L versus M signal. So here we can see that the data do vary somewhat according to the model prediction, but the unique yellow measurements are less extreme than what's predicted by the model. And at this point, it's important to note that while the OCT classification technique that was used for the cone classifications here is highly sensitive, just one misclassified cone can really throw off the percent L estimate quite a bit, since the total of number of cones sampling each of these stimuli is only about 4 or 5. And then, of course, we may have additional errors just from our transfer of the cone locations between the images from the OCT system at Washington and then the AOSLO images we use here at Berkeley. So we're continuing to work with them to assign statistical certainty to each of these cones individually, and perhaps have an additional error metric in the horizontal dimension. So we next tried blending these two models, where the expected L versus M signal was somewhere between the local and the global expectations. And we found that a model that weighted the local expectation by about 78% and the global by 22% fit the data best. So at this point, one interpretation of our data is that local compensation does reduce variability in unique yellow estimates to be less extreme than you'd expect just from a global compensation scenario alone. And one way these results could arise is if the retinal area that the visual system is defining the expected L minus M signal for stimuli around 580 nanometers is actually slightly larger than the stimuli that we've used here in this study so far. So increasing this compensation area, I guess we could call it, or the area over which you would calculate the expected L minus M signal, increasing that inevitably tends towards this global average or the global compensation prediction. And that's because the proportion of L to M cones as you increase the area reflects that of the whole mosaic as more cones are recruited into the sampling scheme. So this led to our next question, which is whether or not we can predict unique yellow for slightly larger stimulus sizes. So in this next set of experiments, the methods were the same as I showed previously, except a range of stimulus sizes were used, and stimuli were organized in this same grid pattern so as to sample the same cones as in the previous experiment. So we tested 8 4x4 arc-minute stimuli, 3 6.5 by 6.5 arc-minute stimuli, and then 1 stimulus that covered the full area of this grid, encompassing all of the cones that were sampled by the smaller 2x2 arc-minute stimuli. And then, as I mentioned previously, we also measured unique yellow for stimuli that were about 1 by 1 degree, which covered the full raster scan of the AOSLO system. So first, and to remind you just of our previous results, we saw substantial variability in unique yellow estimates with the 2.2 diameter stimuli. And here we can compare these measurements to the 2 larger sized conditions where only 1 measurement was made in the 6.5 by 11 arc-minute size and the 1 by 1 degree measurements for both of these subjects. And here I'm showing the frequency histograms for the 4.36 and 6.5 diameter square stimuli, showing that there is still some variability at these sizes in unique yellow estimates, though it is much reduced compared to the smaller stimulus size. So we next wondered whether the average unique yellow of the smaller stimuli would predict the unique yellow estimates for larger stimuli that sampled the same cone photoreceptors. So if we took the average of the 4 unique yellow estimates for the 2.2 arc-minute stimuli, would it predict the unique yellow estimate for the larger stimulus that's spatially coincident in the individual's cone array? And we did find that the average model predictions were significantly correlated with measurements from the experiments. But one observation here that I want to point out is that the model prediction that's shown along the y-axis seems to consistently be overestimating unique yellow compared to the experiment measurements shown along the y-axis. So the opposite was true for a few of these locations and 1 subject, but overall this trend was consistent across all locations in all 3 subjects. So this shows that while the simple average model does pretty well in describing the data, the predictions are consistently overestimating unique yellow compared to the measurements. And this is actually consistent with previous observations by Otake and Cicerone, who showed that unique yellow shifted to longer wavelengths when foveal stimuli were reduced to 3 arc-minutes in diameter. So their result is one that we have recently confirmed using AO-corrected unstabilized stimuli in the foveal center. And this was from work that was presented as an ARVO poster at the 2020 meeting. So just to summarize, unique yellow estimates vary with the local spectral topography of the cone mosaic when stimuli are small and spatially restricted. Larger sizes, variation is still present, but it's reduced. Finally, the variation in unique yellow for larger stimuli is roughly predicted by the average of these measurements for spatially coincident but smaller stimuli. And to conclude, our results are consistent with some local compensation for spectral variations in cone topography, but not enough to make color appearance consistent across local cone neighborhoods with different spectral demographics. And just before finishing, I'd like to acknowledge my co-authors on this work at Berkeley, John Eric Vanston, Austin Rorta, and Will Tooten, and also our collaborators at the University of Washington in the Sabesan lab, including Ram, Vimal, Mary, and Sierra. And I'd like to just finish by acknowledging the funding sources supporting this work, and thank you all, of course, for your attention today.